Hello everyone and welcome to another recorded lecture of BIO1 online. Today we will discuss chapter 3, which is on cells. And as we said before, all organisms are composed of cells, and a cell is the smallest unit of life that can function on its own. And if you're not made of cells, you are not alive, right? Because being composed of cells is one of the most basic characteristics of life. But we didn't know this until microscopes were invented. So that wasn't until the 1600s. And you can imagine that there were many misleading theories about what living things really were until we had a microscope to really look at cells up close. So this might sound a little unbelievable, but people really thought that mice came from dirty shirts and wheat. They thought that a living organism like a mouse can be generated spontaneously from non-living matter. Because again, this is based on observations. I guess people continually saw mice coming out of their dirty clothes, and if you leave some food, the mice will be even more attracted to it. So a really famous experiment was done in 1859 by Louis Pasteur that disproved spontaneous generation. He disproved the idea that living things could come from non-living things. Because another theory was that flies came from meat. So he took 3 pieces of meat in 3 separate flasks and left one open, one sealed, and one that was with transparent gauze. And what he saw was that the flies did not come from the meat. They were just attracted to the meat because when the flask was sealed, no flies came from the meat. Right. So this disproved spontaneous generation and led the way to develop cell theory. So cell theory was first developed in the 1800s, well into the use of microscopy. So we had 100 years to play around with microscopes since Leeuwenhoek discovered it. And now we're studying cells up close. So in the 1800s, we have scientists coming together that come up with cell theory that explains the commonalities of all cells. So notably, the German botanist Schleiden, the zoologist Schwann, and the physician Virchow all established cell theory. And cell theory says the following: all organisms are composed of one or more cells. The cell is the fundamental unit of life. And all cells come from pre-existing cells, right? Cells can't just come from non-living matter. We now add on a couple of new ideas that all cells have the same basic chemical composition, right? They all have the same building blocks. All cells use energy in the form of ATP, and all cells contain deoxyribonucleic acid, DNA, that is duplicated and passed on as each cell divides. So it's important that you understand these components of cell theory. And I'd like for you to, again, you hopefully have done the reading already, but look again into the reading because it talks a little bit about the history of cell theory and how it came to be. It's very interesting. So how we study cells is through microscopes. So microscopy deals with the use of microscopes, and we have different kinds of microscopes to view different types of things. So a light microscope is the one we would use in lab. And that's the one to use when you want to look at an entire cell. Electron microscopes we would not use in lab. Those are very expensive. And those are used to view parts of cells, to really zoom in. And you could even look at viruses. So that would require a lot higher magnification. So magnification is the ratio between the size of an image and its actual size. And an electron microscope can magnify objects hundreds of times more than a compound light microscope. So a compound light microscope can magnify up to about 1,600 times and resolve objects that are about 200 nanometers apart. So that's pretty good. But if you really want to look at a part of a cell or a virus, you need to use an electron microscope. Resolution is the minimum distance between 2 objects that allows them to be seen as 2 separate objects. So basically a microscope with poor resolution might have you see one cell, but if you used that same sample and looked at a microscope with better resolution, you would really see that it was 2 cells. You would be able to resolve that there are really 2 cells there and not just one. And finally, contrast is the difference in shading of an object compared to its background. And sometimes we have to fluorescently label molecules with special colors so they could stand out from their background. So we could actually visualize specific proteins by using fluorescently tagged antibodies that can glow a specific color when they recognize a protein we want to visualize. And you can take a look at these images here that show the combination of low contrast and high resolution, then high contrast but low resolution, and then high contrast, high resolution. So you should be familiar with these terms. So light microscopes are very useful, but their main disadvantage is that we cannot magnify enough. We wouldn't be able to see really the cell structures that we're going to study in this course, for example. So TEM, a transmission electron microscope, is used to visualize internal cell structures. This works by transmitting electrons right through cells. So it sends a beam of electrons through a very thin slice of a specimen, and it uses an electromagnetic objective lens to kind of focus. And what this can do is image a cross-section. So you have to get a slice of a specimen. And TEM, again, is going to bombard the specimen with electrons, and the microscope will translate the contrast in electron transmission into a two-dimensional image that will show the internal features of that object. And this can resolve objects 500,000 times better than the human eye. And it can magnify up to 50 million times. So this is a technique that you would use when you want to look at a cross-section, if you want to look through a cell. The other type of electron microscope is called a scanning electron microscope, and that reveals details on the cell surface. And for this, it's a little different. You have to spray the specimen with a thin coat of metal, and then you bombard it with electrons. And the electrons bounce off of the surface, and the emitted electrons are detected and focused to extrapolate what that 3D specimen might have looked like. So this is going to give you a 3D image of the surfaces. You can remember S for surface. TEM gives you a transverse section, like a cross section. So that's the difference between SEM and TEM. So light microscopes allow us to view living cells, unlike electron microscopes. They transmit light, not electrons. So over here, this is what you would see if you wanted to look at a light microscope for a paramecium. This is what it would look like under a light microscope compared to what you would see. This is a paramecium or Euglena under a transmission microscope. So let's look at the three comparisons. You have over here Euglena under a light microscope. You can see them, they're alive, right? Here under a transmission electron microscope, TEM, you can see the internal structures. With SEM, you can see the 3D surfaces. So prokaryotes, right, cells that do not have a nucleus, like bacteria and Archaea, are 10 times smaller in diameter than most plant and animal cells. So most bacteria and Archaea are about 1 to 10 micrometers. A micrometer is a millionth of a meter. Most plant and animal cells are about 10 to 100 micrometers. Most plant and animal cells are about there. The average skin cell is about like 15 to 20 micrometers. A frog egg is very large for a cell. You can actually visualize that with your own eye. So cells have many different sizes. And to illustrate that, I want to show you a little activity that you can check out on your own over here. So I'm going to navigate to this site right now. So I'll do this really quickly, but you can pretend that this is Times New Roman font, size 12. Here's a coffee bean, here's a grain of rice, and here's a sesame seed. So try to get that size and scale in your head. And now what I'm going to do is gradually zoom in. So now these are still millimeters. You can still see this with our own eye. You could even see a grain of salt against a dark background. But this is where we can't see anything with our own eye. And now, again, single cells. These are individual cells. And here's a human egg. And here's a skin cell. And here's a red blood cell. And now we're going even smaller. So now we have an X chromosome. And now to visualize these guys, you would need to use an electron microscope. Right? See mitochondria, lysosome, E. coli. And then you have viruses, which are even smaller. So you have HIV, influenza, hepatitis. And then we have structures within a cell. So like a ribosome, which is made of proteins, and tRNA, which is a nucleic acid, and other proteins. And then finally, we have the building blocks of life, like a sugar, a nucleotide, an amino acid, and then finally a carbon atom is the smallest. And now I'll just zoom out. How small we're talking about here. All right, so that's a little idea of cell size and scale. So a question becomes, why are cells so small? And it turns out that cells need a large surface area relative to their volume to adequately exchange materials. So we need a large surface area of plasma membrane so they can exchange materials. Like they need to get nutrients in, they need to get wastes out. Right? So the surface area to volume ratio requires that cells be very small. Right? All cells have common features regardless of what kind of organism they come from and where in that organism's body they are. So all cells have these things in common. So make sure you understand this. All cells have DNA, whether you're a bacteria, whether you're a yeast cell, whether you're a plant, an elephant, a human, an oak tree. You have DNA, you have ribosomes, you have a cell membrane, and you have something on the inside of the cell. So whether you are a bacterial cell or a eukaryotic cell, you have DNA, ribosomes that make proteins, cytoplasm, the stuff inside of the cell, and a cell membrane. All cells have to carry out chemical reactions needed to sustain life, right? So all cells need ATP, so all cells have to do cellular respiration. All cells need to make proteins, so all of them will do protein synthesis. We can divide all of life into three domains. Like we said, there's domain bacteria, archaea, and eukarya. And like I said, all cells, regardless of where you are, you have cell membranes, DNA, cytoplasm, and ribosomes. But then we have some distinctions, right? So there's no nucleus present in prokaryotes, but there is a nucleus present only in domain eukarya, right? So there's each domain has its own unique combinations of features. All cells have phospholipid membranes, we said. So let me explain the importance of these membranes. So cells are mostly water and live in aqueous environments. So they need a special membrane that separates them from what's outside. Right? So each cell needs to have a barrier that protects it from the outside world. This barrier, this membrane, consists of two layers of phospholipids. Because it consists of two layers of phospholipids, we call it the phospholipid bilayer. And this is one phospholipid. You can see this is a hydrophilic head. It's hydrophilic because it has a polar group and a phosphate. And it has two hydrophobic fatty acid tails. So these are two fatty acid tails that are both nonpolar. And this is a polar hydrophilic head. The way that phospholipids orient each other is that they're all lined up with their heads facing the watery outside or the watery inside, with all the fatty acid tails facing each other. So let's look at this cell. Like this is how the phospholipid bilayer is oriented, with the watery inside and a watery outside. So remember, hydrophilic substances like water. So that's why the hydrophilic polar heads will always be oriented toward the watery outside or the watery inside. And the fatty acid tails are hydrophobic, so they want to be hidden. So they want to be buried in between. Right, so we said the cell membrane or the plasma membrane, I'll use those interchangeably, they function as a selective barrier. So it's selectively permeable. It only allows certain things in or out. Specifically, right, it's going to want to let oxygen and nutrients in and wastes out. But otherwise, it has to be very controlling about which substances can enter. So in general, lipids and small nonpolar molecules have no problem getting in and out of the cell. But if you're polar, a large molecule, or charged, like an ion, you're going to need help from a special protein. Right? So small hydrophobic molecules have no problem getting inside of the cell, but hydrophilic molecules need help from membrane proteins to enter the cell. There's some exceptions, like very, very small hydrophilic molecules can sometimes sneak in between the phospholipids, but for the most part, hydrophilic molecules like sugars or amino acids need help from membrane proteins to enter the cell. So remember, this is just the same thing I showed you before, but I can compare this to a bread and butter sandwich, where these fatty acid tails are kind of this buttery interior, and the bread can absorb water. That's hydrophilic. So how do hydrophilic substances enter the cell freely? Right? So for example, suppose I have this watery dye that I want to get inside of the cell, but this butter is so hydrophobic, it's not going to allow the hydrophilic dye to get through. Right? It's not just going to sit on top. It won't be able to get through this hydrophobic barrier to the other side of the cell. So we need to use membrane proteins. And membrane proteins help transmit information and materials from the outside of the cell inward. There are many different membrane proteins. Each carries different functions. So one example of a membrane protein would be like a channel protein, which is kind of like an olive without a pit that's embedded inside of the bread and butter sandwich. And what this can do, this is like a protein, it will allow the hydrophilic dye to penetrate through the hydrophobic fatty acid tail layer and get inside of the cell. Right? So proteins act as channels within the membrane that allow substances inside. And again, this is just one example of like a transporter protein that's like shown here. But really, we have a lot of different kinds of membrane proteins that are very important for the cell, some of which help cells stick together. Others are receptors that wait for a special message on the outside of the cell to bind so they can tell something to the cell on the inside. So membrane proteins are incredibly important for cell function. And in fact, more than 50% of all medicines that we take bind to membrane proteins to function. So whether it's an antidepressant or an antacid, right? These drugs bind to special proteins that are embedded within your cell membranes and stop them from functioning or help them to function in a certain way. And what I want to try to say is that the membrane is almost like the brain of a cell, right? So oftentimes people learn in high school that the nucleus is like the brain of the cell, but I don't really think that's a good example. Some cells, like red blood cells, don't even have a nucleus, and they're able to function over a month just fine. But we know that a human cannot, or any organism cannot function without a brain. You cannot have a cell without a functional membrane. That's impossible. Every cell needs to have an intact functional membrane in order to survive. So the membrane of the cell is just like our brain of us. And let me try to illustrate this with an example. On our membranes, we have a lot of different proteins like receptors, and these receptors are waiting for certain signals to bind. This signal won't be able to just cross into the cell because the signal is usually polar or too large. So it needs to bind to a receptor. And if that signal bound to a receptor, something on the inside of the cell would change to cause some kind of an effect. So in that way, the cell would know what's going on the outside through this receptor, right? And this can happen like with calcium as well. So when calcium gets inside of the cell, it has to get through a special protein. So this is another membrane protein. It's called a calcium channel. Once this protein allows calcium in, you're going to see a lot of stuff going on the inside. All right, so you don't have to memorize this, but in response to calcium entering the cell, that activates this protein called PKC. And that will activate this protein, which will activate that protein, which will activate this protein, which will activate that protein, which will activate this protein, and that will finally tell something to go inside of the nucleus and cause a change in response to this calcium binding. Again, how would this cell have known what to do, right? It needed calcium to tell it what to do, but calcium couldn't just go inside and cross the membrane, right? It needed to go through a special protein, a special membrane protein. And this is another kind of receptor protein that's waiting for a special signal on the outside to bind to it. And once that signal on the outside binds to this receptor, things on the inside of the cell, inside of the cytoplasm, can be activated. So how is this like the brain? What our brain does is it perceives, it waits for signals from the outside world, right? It's a receptor, it's a receiver that's waiting for signals, whether they are scents or sounds or sights. And what our brain does is it interprets those signals. When it gets those signals, the inside of the brain has all these pathways, all these neural networks that have to be processed. So we understand what we're actually seeing. The brain at the same time acts like the membrane because it blocks out anything we don't need. Right now, you're focusing hopefully on this lecture and not on the beeping cars outside or your screaming kid next door, because your brain is filtering that stuff out. Your brain doesn't need to have that input, right? Your brain needs to focus so you do well on the exam. So the brain, right, is a receiver. It receives signals, and it's a filter. Same idea of the cell membrane. It's a receiver and it's a filter. Another aspect that's really important of the cell membrane are the carbohydrates. So there are chains of sugars that are attached to some of the proteins and some of the phospholipids themselves in the cell membrane. So some of these proteins could be glycosylated. And these help cells recognize each other. And also, each one of your cells has a very specific decoration of sugars that only identical twins share in common. So there's a way for cells to recognize each other and to distinguish themselves between other. Also, there is cholesterol, which we know is another type of lipid, right? It's 4 fused rings. And these help keep the membrane at the right level of fluidity. We want to make sure they're not too rigid and not too fluid. So cholesterol is one example of a membrane steroid that helps the membrane stay at the right consistency, so to speak. So remember, this creates a consistency that's more like a jello. It's not a liquid membrane. It's not a solid membrane, right? And because a cell membrane consists not only of these phospholipids, but also these proteins and these sugar molecules, we call the whole membrane a fluid mosaic. It's composed of a lot of different parts, but they also are a fluid because they drift. So these phospholipids drift. They're very much fluid, right? They don't just stay in one place. Sometimes they even flip places. Right? There are certain enzymes that can help flip them around. So a mosaic is just static, but a membrane is a fluid mosaic. So here's a quick video on the lipid bilayer that I'd like you to watch now.
These materials move in and out of cells by passing through the plasma membrane of the cell. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of a lipid bilayer. Phospholipids make up a large part of the membrane and form a bilayer. The structure of the bilayer is due to the tail-to-tail packing of the nonpolar hydrophobic tails, composed of 2 fatty acid chains, and the polar hydrophilic heads, composed of glycerol and phosphorylated alcohol. The lipid bilayer is 5 to 10 nanometers thick and is embedded with proteins. Some cell membranes also contain cholesterol. A plasma membrane contains different types of proteins which are specific to the particular function of the cell. These proteins also enable the cell to interact with its environment. The entire structure of the plasma membrane can be described as a fluid mosaic model. The phospholipid bilayer has properties resembling fluids, and the differing proteins and their attachments on either side of the membrane resemble a mosaic.
So pause here and try to answer this question. And the answer is D. And please pause here and answer this question now. The answer is A. So now another thing that all cells have in common are ribosomes. And ribosomes are cellular machines that make proteins. Right? The ribosome itself is made up of ribosomal RNA, a type of nucleic acid called rRNA, and other proteins. So the ribosome consists of two subunits. Their subunits are both made up of rRNA and ribosomal proteins. Ribosomes are either found floating in the cell or attached to the rough endoplasmic reticulum, or the RER, of eukaryotes. So if you're a bacteria cell or an Archaea cell, you don't have an endoplasmic reticulum, so your ribosomes are just floating around. But if you're a eukaryote and you do have an endoplasmic reticulum, then your ribosomes are attached to it. And that's what makes this endoplasmic reticulum appear rough. We'll talk about that later. So before we go on to eukaryotic cells, let's just finish with bacterial cells. So bacteria are prokaryotic, meaning they do not have organelles. They do not have a nucleus. Their ribosomes and their DNA kind of just float around. There's no organization. Everything happens in this one little cell. Bacteria and Archaea are each single-celled, right? As simple as can be. These are the earliest life forms. The DNA is located in a nucleoid region, but again, there's no separate membrane surrounding the DNA. That's why we call this a prokaryote. A eukaryote has a membrane surrounding its DNA. In addition, bacteria have a special cell wall made of peptidoglycans, which is like a combination of protein and sugar, and that's outside of their cell membrane. So they have a cell membrane, and then they have a cell wall. Outside of the cell wall, they may have a capsule in addition, and many bacteria require flagella, which are made up of microtubules, as we'll soon see, that helps propel them, helps them give them motility. Bacteria come in three main shapes. They can be rod-shaped, they can be spherical-shaped, or they can be spiral-shaped. And recall that bacteria are usually about 1 micrometer in diameter. Between 1 to 10 micrometers is the general rule of thumb. So that's all we'll really talk about with bacteria for now. We're going to focus a lot more on eukaryotic cells, like animals, fungi, and plant cells. And in eukaryotic cells, DNA is contained within a nucleus. They also have many different organelles, right, many organs that are separated by membranes. And this is what allows for a lot of complexity, right? If you have separate compartments to do different things, you can divide the labor, right? So each little compartment has its own little, its own separate structure, which means it has its own separate function. So it's like if you have a studio apartment, you have your kitchen, your living room, your laundry room all at once. It's not as efficient. If you have a big house, you can separate. So you can have all of your laundry equipment in one room, and then all of your cooking equipment in another room, and then all of your leisure stuff in another room, all of your gym stuff in another. So by dividing things up in separate rooms, you can actually be a lot more efficient and a lot less cluttered. That's what eukaryotic cells do better than prokaryotic cells, right? They can divide and they can specialize a lot more. And again, it's all because of these membranes, because membranes surround each of these organelles that allow them to keep their own internal environments just specific for their function. So now I want you to really pay special attention to the difference between animal cells, plant cells, and bacteria cells. You should be able to understand within your notes what animal cells, plant cells, and bacteria cells have in common. So by making a Venn diagram, it's really helpful. And you would put in everything like in this overlap that animal cells and bacteria have in common. Over here in this little cross section is what all three have in common. And here be just plant cells, for example. You could also do that in a table by coming up with each cell structure and then say where it is in the cell, what the function is, some notes about the structure, and whether it's found in bacteria, plants, or animals. Also, as you go through this lecture, it's kind of interesting to think about eukaryotic cells as miniature humans. If you were to just take a moment right now to pause, I'd like you to just come up with the major functions of the human body. So literally just take 20 seconds and write down what our human bodies need to do in order to function. So pause here. Here's a list that I came up with. And it turns out that for every single thing that we need to do, whether it's eat, move, get energy, digest, grow, die, reproduce, evolve, protect, or communicate, the cell has to do as well. And the cell does that with a specific organelle or structure. So you can take a second to again pause and really come up with the analogies, because for everything that a human needs to do, a cell needs to do on a smaller scale. So you can pause here and answer this quick question before we move forward. All right, the answer is A. So eukaryotic cytoplasm consists of organelles and cytosol. So the cytoplasm refers to everything inside of the cell other than the nucleus. The cytosol is shown here. It's a fluid. It's like a gel of small molecules and proteins. So the cytosol, again, we would have the cytosol also found in bacteria cells. But what we don't have in bacteria cells are all these organelles. We only see organelles in eukaryotes. And we said each organelle is protected by phospholipid membranes that allows it to keep its own environment to allow them to do their own functions. We'll talk about the endomembrane system first, which are organelles that transport materials within the cell and out of the cell. That'll be the first category. Those include the endoplasmic reticulum, the Golgi, vesicles, and more. Also, organelles that break down waste are included in the endomembrane system. So lysosomes are part of the endomembrane system. Microbodies are organelles that have specialized enzymes that help perform specific functions. So we'll talk about peroxisomes and vacuoles. And finally, we'll talk about energy-related organelles that capture or store energy, like mitochondria and chloroplasts. Right? And these guys are independent and self-sufficient on their own. So DNA specifies the recipe for the proteins in prokaryotes and eukaryotes. And prokaryotes just have a single circular chromosome of DNA that is not enclosed by a separate membrane, right? However, eukaryotic cells store DNA in the nucleus. So plants, animals, and fungi store DNA in the nucleus, and a nuclear membrane protects the nucleus, but has nuclear pores in the membrane to allow materials in and out. So true or false, every nucleus in your body has the same DNA. So take a second to think about it. We can pause here. And this is actually true. So your muscle cell nuclei and your eye cell nuclei and your nerve cell nuclei and your skin cell nuclei all have the same genes. And you should be asking the question, how does a skin cell differ from a muscle cell then? If they have the same genes, why are they so different? And we're going to answer that question later in the semester. But every nucleus has the same genes, but not all the genes get expressed into proteins. And that, again, will become a lot more relevant later on. So here's the nucleus in a typical animal cell. The inside of the nucleus is called the nucleoplasm. Ribosomes are built in the nucleus within a portion called the nucleolus. And again, the nuclear membrane is made up of a phospholipid bilayer, just like the cell membrane. And here are those little pores that allow things in and out. DNA is stored in the nucleus as chromatin, and chromatin is DNA associated with other proteins. So now I want to introduce the central dogma of biology, which we'll talk a lot more about later in the semester, but I want to give it a brief introduction. And the central dogma of biology begins in the nucleus. Like I said, the nucleus contains DNA, which specifies the recipes for the proteins needed in the entire organism. And the central dogma of biology states that DNA has the instructions to make proteins. So this all begins in the nucleus because the DNA is stored in the nucleus. So let's give an example of mammary glands. So special cells in the mammary glands of female mammals produce milk, which contains proteins, fats, carbohydrates, water, antibodies, other really specialized proteins, other minerals like calcium and potassium. So The milk-producing cells of the mammary glands of a female are dormant most of the time. But shortly after the female gives birth, there's a burst of productivity. And we're going to give this example. Let's say this is one mammary cell. How does the milk proteins and the milk fats get packaged and delivered to the milk ducts, right? So the nucleus, right, the process of milk production and secretion begins in the nucleus because that has the recipe for all the milk proteins and all of the enzymes required to make carbohydrates and lipids. So the first thing that has to happen is that the DNA recipe is rewritten into messenger RNA, which is called mRNA. The mRNA molecule then leaves the nuclear pore into the cytoplasm. So the mRNA, which is a copy of the DNA recipe, leaves the nucleus. Then the ribosomes are the next in line. The ribosomes follow the mRNA recipe to make proteins for the cell. And like I said, some proteins are made on ribosomes in the cytoplasm that are kind of floating around. Others are made on ribosomes attached to the endoplasmic reticulum. So here's a ribosome associated with the rough endoplasmic reticulum. And you can see the ribosome is right here where I'm pointing. The ribosome is reading the mRNA recipe and is going to be making proteins from that recipe. The rest of these organelles are going to modify and sort these proteins into vesicles. A vesicle is a little bubble of membrane, which can then be transported out of the cell. The endomembrane system organelles will modify and sort the proteins in vesicles, which will be, again, delivered to the milk ducts, so the baby can then eat. So next up, we said the endoplasmic reticulum, were a network of membranes that border the nucleus and extend throughout the cell. And if the endoplasmic reticulum has ribosomes on it, we call it the rough endoplasmic reticulum. The rough endoplasmic reticulum is the site of protein synthesis. But in addition, also some proteins get modified in the endoplasmic reticulum. So the rough endoplasmic reticulum is right outside of the nucleus, and that's where proteins are modified and folded into their exact 3D shape. Right? So the endoplasmic reticulum helps the proteins form into their proper shape, their proper structure. The smooth endoplasmic reticulum is adjacent to the rough endoplasmic reticulum, but makes lipids. Doesn't make proteins, it makes lipids. So for example, milk has a lot of fats in it. So the smooth endoplasmic reticulum will make the lipids required for milk. The rough endoplasmic reticulum will make the proteins required for milk. The endoplasmic reticulum is also the site of detoxification. So it can actually detoxify drugs and poisons. Proteins from the rough endoplasmic reticulum and lipids from the smooth endoplasmic reticulum are packaged into vesicles, which are little bubbles of membrane. Okay. So we said the ribosomes are what are going to make the proteins, and some of them could be attached to the rough endoplasmic reticulum. That's shown here. So here's the rough endoplasmic reticulum, and here you have a ribosome that's reading the mRNA recipe, and that is making a protein. And that protein is consisting of amino acids. So those amino acids get translocated inside of the endoplasmic reticulum, where they can then fold into their proper shape. You can also have ribosomes that are kind of free-floating in the cytosol. And those ribosomes synthesize proteins that will only function in the cytosol, right? The ribosomes that we're talking about before that are attached to the rough endoplasmic reticulum are going to make proteins that function inside of organelles or outside of the cell. So again, this is an important distinction. We said proteins are made by ribosomes, but ribosomes could be found in the cytosol, or they could be attached to the endoplasmic reticulum. If a protein is destined to just be inside of the cytoplasm and just hang out, then it could be made on a free ribosome like this. If the protein is destined to be secreted outside of the cell, or destined for a specific organelle, then that protein is made by a ribosome on the rough endoplasmic reticulum. So that's an important distinction. So where are ribosomes found? You can pause here. And the answer is D, B, and C. So what we said so far, we have these purple proteins were made by the ribosomes on the endoplasmic reticulum. The smooth endoplasmic reticulum has enzymes that can manufacture lipids, which are the yellow spheres. Right? So far, we're up to this step over here. So the next step is the vesicles that leave the endoplasmic reticulum containing lipids and proteins fuse with the Golgi apparatus, which is a sac of membrane sacs that acts as a processing center. So the Golgi apparatus is a different organelle that can help process and sort the vesicles to where they need to be. Right? So in the Golgi apparatus, the proteins and lipids are processed and packaged for export out of the cell. So this is a better view of the Golgi apparatus, and incoming vesicles from the endoplasmic reticulum would join this side. And in here, in these flattened sacs called cisternae, these proteins would be modified. So different enzymes could help make these proteins fully functional. And then finally, after they're done, they'll be ready to go to an organelle or outside of a cell. So what the Golgi apparatus is doing is it's not only modifying the proteins made on the rough endoplasmic reticulum, but it's directing them to their proper destination. Right? So the Golgi apparatus sorts vesicles for shipment, kind of like a postwoman would do. If you're in a mailroom, you have a lot of different envelopes with different zip codes, and you have to look at each envelope and see where each envelope belongs, right? And that's based on a zip code. The Golgi apparatus does just that, right? It looks at all the incoming vesicles, and then it tells them where they belong. Instead of a zip code, each vesicle has special sugars or proteins on the outside of it that tell the Golgi apparatus where its destination is. So the vesicles are either exported from the cell, we call that secretion, or exocytosis. Exo is outside, cyto is the cell. The vesicles could be transported to organelles within the cell, or they could be merged with the plasma membrane. Suppose it's a protein, it's a membrane protein that's supposed to be stuck in the plasma membrane, right? So that will be, the Golgi will recognize that, and then it will tell this vesicle, okay, you're supposed to fuse with the plasma membrane and stay there. So that's what the Golgi apparatus is doing. So finally, we said the Golgi packaged and processed them. So the Golgi apparatus realized, right, so to speak, that there were milk proteins and milk fats in these vesicles, and that absolutely belongs to be secreted from the cell. So these vesicles, these transport vesicles, are released from the cell when they fuse with the cell membrane. And then all the proteins and lipids can be brought to the milk ducts, where the baby can then be fed. So that's the end of the central dogma of biology and exocytosis bit. So now we'll talk about lysosomes. Lysosomes contain enzymes that break down cellular components by hydrolysis. So lysosomes are almost like the garbage disposal of the cell. And these are mostly found in animal cells, right? Plant cells do not have lysosomes for the most part. There are some exceptions. But again, lysosomes are mostly found in animal cells, and any vesicle that has waste in it will be sent to the lysosome. So for example, over here, you have a vesicle that has a partly digested has a damaged mitochondria. So what the lysosome is going to do, it's going to engulf the mitochondria and break it down. It's going to digest it like a garbage disposal, then chop it up into pieces. Also, the lysosome can break down debris from the outside of the cell. So debris can be internalized by the cell. That's called endocytosis. And the lysosome can then digest it and release the end products to be recycled. The lysosome requires a very acidic environment. So very low pH is really important. Kind of like the stomach requires a lot of stomach acid. So does the lysosome require a lot of acid. And Tay-Sachs is a genetic disorder in which the enzyme hexoaminidase A is inactive. So normally our lysosomes have these enzymes that break down any fatty acids that we don't need anymore or that accumulate, right? It breaks down any garbage so we don't get backed up. However, you might have a genetic mutation in which the instructions for one of the enzymes is incorrect. So you can't make the right enzyme. Therefore, you're going to get an accumulation of all these fatty acids. All these fats are going to accumulate. And that's what happens in the neurons of people with Tay-Sachs disorder. So unfortunately, it's a very debilitating disease, and it's a genetic disorder that can be detected at birth or now prior to birth, actually, with a lot of genetic techniques we have. But again, this is one reason why the lysosome is so important, right? The lysosome breaks down anything so the waste doesn't accumulate, right? It can also get rid of any damaged organelles or any cell debris. Lysosomes are essentially made by the Golgi as part of the endomembrane system. And this can be a little complicated to understand, but remember that lysosomes are just packages of enzymes, and enzymes are proteins. So those enzymes had to have been made by ribosomes on the endoplasmic reticulum. They were just packaged in special membranes, and then these are called lysosomes once they're fully mature and packed with enzymes that can do hydrolysis, right? They break down cellular components. So here's a picture of a lysosome that actually has an entire organelle within it. All right, here's a mitochondrion, and a peroxisome within one lysosome. So this is called autophagy. The lysosome can eat part of itself and damage it and break it down so it can be recycled. So you can watch the video on lysosomes that's contained within the ebook on Connect. So let's just summarize the endomembrane system very quickly. We said the rough endoplasmic reticulum synthesizes proteins and packages them into vesicles, which then go on to the Golgi apparatus. Right? We also know that the smooth endoplasmic reticulum makes lipids, which will also go to the Golgi apparatus. The Golgi then modifies lipids and proteins from the endoplasmic reticula, sorts them and packages them into vesicles. The vesicles, if they're destined to be secreted, would fuse with the plasma membrane, and that's called exocytosis. You could also have any debris be digested by the lysosome. So this is called endocytosis. The cell can consume garbage from the outside. The lysosome containing digestive enzymes will then break down that substance so it could be recycled. All right, you could just pause here and make sure you understand what I just said. This is the breakdown. Right? And the key of the endomembrane system is that organelles can only interact because their membranes can fuse together. So all these membranes are fusing and then unfusing with each other. So that's a way that they can all work together. Right? And it's the special proteins and carbohydrates on organelles and the vesicles that allow them to communicate with each other. So if you're going to go on in your study of cell biology, you'll learn a lot more about how these communications occur within a cell. The next organelle we'll talk about are peroxisomes. And all eukaryotic cells contain peroxisomes that have, again, a very similar function to lysosomes because they break down lipids. They also detoxify molecules. So the peroxisomes are similar to size in lysosomes and have a similar structure. But again, they metabolize lipids specifically, and they detoxify poisons. So peroxisomes are almost like the liver, right? Like the liver in your body detoxifies poisons. It helps you metabolize lipids. Same idea as the peroxisome. It's called the peroxisome because it catalyzes reactions that produce hydrogen peroxide. And unfortunately, peroxide is a toxic substance that can damage DNA. So the peroxisome also has to make catalase, which is an enzyme that breaks down the toxic peroxide into harmless water and oxygen. So that's important to know because we don't want the peroxide because it can make hydrogen, it can make free radicals that can damage DNA very easily. And that's a reason why you shouldn't use hydrogen peroxide on any of your wounds. Right? It's better to use soap and water and not hydrogen peroxide as was once thought. Defective peroxisomes may lead to severe mental impairments in humans. One such impairment is called ALD, or adrenoleukodystrophy. And that's a disorder that damages the white matter of the brain and also impairs the adrenal glands. And again, it's when you have an accumulation of certain waste products, right, you can't have your organs function properly. So peroxisomes are required to break down any waste materials, any waste fats specifically, and any poisons. So the peroxisomes are detoxifying, especially in the liver and the kidney cells, and they're breaking down lipids. Right. So now we'll talk a little bit about a plant cell. So plant cells are also eukaryotic. They have the same membrane-bound organelles as animal cells, but a few notable differences. Right. So they have a large central vacuole, and they also have chloroplasts, which we'll talk about, and they have a special cell wall. So plant cells have large central vacuoles. So unlike a lysosome in an animal cell, right, plant cells rely on vacuoles. As cellular digestion occurs here, and vacuoles also help regulate the size and the water balance of plant cells. The large central vacuole regulates turgor pressure in plants. What that means is the vacuole needs to be filled with fluid so it can help the membrane press against the cell wall, which is a very firm structure. And when the vacuole is filled with water, then the plant can be upright. When the vacuole is empty, that's when the plants would be limp. So this is why it's important to water your plants. Right? When you water your plants, water can be delivered from the roots through the plant cells, and the water will be stored in the vacuole. Mitochondria, the next organelle we'll talk about, are what generates cellular fuel, and plants and animal cells have many, many mitochondria. Mitochondria themselves have two membranes. The outer membrane allows small molecules to pass. The inner membrane is the site of ATP production, which is the main source of cellular energy. Right? So we need energy for a cell to grow, divide, produce proteins, and do chemical reactions. So we need lots of ATP, which means we need lots of mitochondria. The inner membrane is folded into what is called cristae. So that cristae adds tremendous surface area to the inner membrane, which can help us make the maximum amount of ATP in the tiniest space. So we want to fold the cristae over and over again because it's packed with enzymes that can make ATP. Inside of the mitochondrial membrane is called the matrix, and that contains important enzymes that are needed to convert glucose into energy in cellular respiration. It's important to note that mitochondria are maternally inherited. So in most mammals, mitochondria are inherited from the female parent only. That's because the mitochondria in a sperm cell degenerates before, as it matures. The mitochondria actually becomes part of like the motor system that can propel the sperm's tail. So that's another story. I would go more in detail in class. But the mitochondrial DNA, again, is useful for tracking inheritance through female lines because we only get it from our mother. It's also important to know that we have separate DNA, right? Mitochondria have their own genome, their own DNA, and they divide separately from the cell. So they have their own division and their own genome. So it's true that there is DNA outside of the nucleus found in the mitochondria. Right? So of course, since mitochondria have their own proteins, they have their own ribosomes and DNA. And we'll talk about this very soon. So cell respiration is the process that produces ATP. So cell respiration in the mitochondria extracts energy from food, and it could convert that energy from glucose into ATP. Mitochondria produce ATP by adding a phosphate group to ADP via a high energy bond. And I mentioned that in the last lecture. Right, so ATP is adenosine triphosphate, and it stores tons of energy in this last bond. So what the mitochondria is doing, it's basically using the energy from the glucose that we have in our food to tack on an extra phosphate onto ADP, forming ATP. Once we have ATP, we could then break off a phosphate to harvest that energy. Again, we'll go into a lot more detail when we talk about cellular respiration, but this is what's going on in the mitochondria. So where's the most energy stored in ATP? You could pause here. So the answer is B, right, in the bond between the last two phosphates. So now we'll talk about chloroplasts. So plants and algae contain chloroplasts because they need to do photosynthesis, right? So photosynthesis occurs in chloroplasts, right? And chloroplasts have a permeable outer membrane and an impermeable inner membrane, just like the mitochondria. But these are actually a lot larger than a mitochondria. And the thylakoids, so thylakoids are these little discs. Each disc is called a thylakoid. And thylakoids have an abundance of chlorophyll. It's a pigment called chlorophyll that can convert sunlight energy into sugars. All right, that's what photosynthesis is, right? We said plants just have to sit outside in the sun and they can make their own sugars. Well, the chloroplast is where it happens. The chloroplast is almost like a solar panel, right, that can absorb the sun's energy and use it to then make sugar in photosynthesis. So these are the solar panels of a cell. And these solar panels, like the thylakoids specifically, thylakoids are stacked in grana. One stack is called a granum. Singular is a granum, but you see these stacks of thylakoids. The surrounding space is called the stroma, and that's where the sugars get produced. A chloroplast is one example of a larger category of plant organelles called plastids. So some plastids synthesize lipid-soluble pigments like red, orange, and yellow carotenoid pigments, like those that you'd find in tomatoes or carrots. But a plastid, for example, is a chloroplast, right, that do photosynthesis using chlorophyll. So chlorophyll, again, is what makes plants appear green. And we'll talk a lot more about that when we discuss photosynthesis. Where does the sugar go on next after it's made in the chloroplast? So that's a good question. So after the chloroplast makes sugar from the sunlight, where does that glucose go? Right? It goes to the mitochondria. Remember, this is a very common misconception. People think like, okay, animal cells have mitochondria and plant cells have chloroplasts. No. All eukaryotes have mitochondria, because all eukaryotes need ATP, right? The difference is only plants have chloroplasts because they could just use the sun to make their sugar. And there, the sugar would then go to the mitochondria to be converted to ATP. We don't have that luxury. We don't have chloroplasts. So we have to eat sugar, and then the sugar could go to the mitochondria. So regardless, plants and animals need sugar that go to the mitochondria to make ATP. Where the sugar comes from is whether you're a consumer or a producer. Just like mitochondria, chloroplasts have their own ribosomes and DNA and have their own cycle of division. And this brings us to the origin of the eukaryotic cell. So fossil records suggest that the first cells were prokaryotes, and that prokaryotes must have gained a nucleus to become eukaryotes by invagination of the plasma membrane. So the membrane must have been internalized and surrounded the DNA. And then those cells must have been better off because they had DNA protected by a membrane. And that invagination process can also explain the origins of the endoplasmic reticula and the Golgi apparatus. The energy organelles, like the mitochondria and chloroplasts, may have originated when eukaryotic cells engulfed smaller prokaryotic cells. So what this suggests is that mitochondria and chloroplasts might have been their own independent organisms that were living on their own, right? So eukaryotic cells would have benefited from the ability to utilize oxygen, right, with the mitochondria, or to synthesize organic food from the sun, like in a chloroplast. So endosymbiotic theory is the name of this hypothesis. And this was very famous by a woman, Lynn Margulis. And this theory was saying that the original cells about 3.8 billion years ago were just prokaryotes. They just had DNA floating around. But about 2 billion years ago, early cells must have gained a nucleus by having some of the membrane surround their DNA. So that nucleus allowed special functions to be assigned, right, which frees up cellular resources for other work. So this is very advantageous. The cell must have gained an endomembrane system by, again, more proliferation of membrane, and that increased surface area would allow for a higher rate of transport of materials within the cell. So that's another huge advantage. Then at another juncture, the cell must have gained mitochondria. And again, aerobic bacteria, these are just individual bacteria. They were doing their own things, and they can make ATP using oxygen. So now, if the cell says, hey, bacteria, do you want a free home? I can protect you. And the bacteria says, sure. In return, I'll make some energy for you. I can make ATP. So now we have a better advantage. Now the cell can metabolize sugars in the presence of oxygen to make tons of ATP. And that is how we get our current animal cells, right? Our animal cells now have a nucleus and a membrane system and a mitochondria. At one point, those cells must have also gained a different photosynthetic bacterium that was able to make its own sugars from sunlight. And those might have been engulfed by certain cells, and those must have become plant cells. So it's called endosymbiosis because they're symbiotic. We're working together. And endo because these bacteria were internalized, right? Endo means internalized. So endosymbiosis talks about how these organelles were once independent bacteria that we work together with. So we are actually a product of other organisms. And in fact, you are made up of tens of trillions of cells, but you have even more cells in you that are not human. So you actually consist of more non-human cells, like bacteria in your gut, than human cells. So you can watch the video contained in the ebook to learn more about endosymbiosis. So a cell wall is something that is not found in animal cells, right? It's found in plant cells, bacteria, and fungi. Cell walls help protect the cell and keep the cell's shape intact, and the thickness and chemical composition of cell walls differ from species to species and among cell types. Plant cell walls are made up of cellulose, which you now know is a polysaccharide made of glucose, right? And that provides very rigid support from the cell. So you can think about the cell as a factory. Right? You could pause here and take a look and try to come up with why I'm putting these names according to this little factory diagram. So you should be able to come up with an analogy of your own even, to come up with a way to remember what each organelle does, what the function of each organelle does, and how they work together. So here's a good image from a different textbook that I thought was pretty helpful, very detailed, tells you everything you need to know without including any unnecessary information. We could organize our organelles by function. We said organelles that store and clean up are vacuoles, lysosomes, and peroxisomes. Organelles that build and sort proteins are ribosomes, endoplasmic reticulum, and Golgi apparatus. We spoke about organelles that capture and release energy, like chloroplasts and mitochondria. And then we spoke about cellular boundaries, like the cell membrane and the cell wall. So this is a way to keep it organized in your head, but again, you don't have to memorize this like in this order. This is another image I scanned from an old textbook that I really like because it talks about each organelle, tells you the function, and then it tells you whether it's in prokaryotes or animals or plant cells. And this is exactly what I want you to understand from this chapter the most. All right, so it's very important to come up with some graphs or tables to help your brain keep this information. And again, this is the same type of thing that tells you structure, function, and what type of cell it's found in. Finally, we're going to talk about the cytoskeleton and molecular motors. So the cytoskeleton, or the cell's skeleton, consists of elaborate arrays of protein fibers and tubules that function in keeping the cell's shape intact, maintaining mechanical strength, It allows cells to move, and it allows things to move inside of the cell as well. So the cytoskeleton is very important, and you can see these are images that were done using a technique called immunofluorescence, where the specific cytoskeletal proteins were labeled with fluorescently labeled antibodies. So you can see specifically here, these are microtubules in green, shown here. In this image, the intermediate filaments are shown in green. So we can study the structure using a lot of these special imaging techniques. So what's the purpose of a skeleton, right? Why do we need a cytoskeleton? Well, why do we need a regular skeleton? Right? What would happen if we didn't have a skeleton? We'd be a blob with no structure, and we'd be unable to move. So that's exactly why a cell needs a skeleton. It needs structure, and it needs to be able to move. But instead of bones, the cytoskeleton is made up of proteins. And there are 3 different types of proteins that are found in the cytoskeleton. The first protein are called microfilaments, and microfilaments are the thinnest. Those are made of actin proteins. Intermediate filaments are intermediate in diameter, and they're composed of protein ropes. Finally, microtubules are the thickest, and they're composed of tubulin subunits, and those are hollow in the center. So we can take a look here. You can see a major component of structure is usually found in actin. All of these proteins help with structure. But you can see like the structure of this car over here is very much due to actin. The peacock's feathers are built of intermediate filaments. So intermediate filaments provide a lot of structure. And these microtubules help this chameleon deliver pigments to its surface to change colors. So microtubules allow transport of things. They're like train tracks, where intermediate filaments are more structural, and actin has a lot of purposes. So let's talk about actin later. Let's start with intermediate filaments. So intermediate filaments function as support within cells and support between cells. So when you think about intermediate filaments, think about strength. They're very strong. This is a sheet of epidermal cells, like skin cells, and they're all joined by intermediate filaments. It keeps them intact in one sheet. Similarly, intermediate filaments are very strong, right? So they're found in claws and feathers and porcupine spikes. So things that require a lot of structure are very rich in intermediate filaments. An example of an intermediate filament is keratin, which you might be familiar with, is a component in hair. So keratin is an example of an intermediate filament. What makes intermediate filaments so strong is that they're bundles of like these protein ropes. So it's almost like a pull and peel Twizzler, where it has these individual strands that are wrapped around each other. So why do we need intermediate filaments? So suppose we have 2 sheets of cells, one that has intermediate filaments connecting adjacent cells, and one without intermediate filaments connecting adjacent cells. Suppose we stretch each of these. What would happen? Well, if we have intermediate filaments, that would keep all the structure intact. It would keep all of them together, so the cells are fine. And that's why our cells are just fine when we move around. However, if you do not have intermediate filaments connecting the cells would rupture when you stretch them. And unfortunately, that manifests itself in a terrible disease called epidermolysis bullosa simplex, which is a hereditary skin disorder due to very short keratins in the skin tissue. So normally keratins help our skin cells stay together. They help them stick together. But what happens if you do not have long enough keratins is that the skin cells will separate from each other. They can't stick together. And when they separate, they form blisters. So even in response to bending over to pick up something, the skin cells of your back may separate because there's nothing connecting them together. Next, we'll talk about actin, or microfilaments. Right? And microfilaments are the thinnest of the 3. They're important for cell strength, cell locomotion, so cell movement. They also play a role in cell division and muscle contraction. Microvilli, shown here, are extensions of the plasma membrane, and actin gives these little protrusions their structure. So sometimes we want to increase the surface area of some cells, and actin allows the membrane to protrude a little bit, forming microvilli, and that again allows more surface area. So in the intestine, we can absorb more nutrients because there's more surface area of membrane around these actin protrusions. Actin also makes pseudopods, which are protrusions made of actin that propel motile cells. And what happens is this is called cell crawling. So cells can crawl because they grow a lot of actin on one end. I'll show you this. So over here, we have a bunch of actin is going to grow on this side, and that's going to push this single-celled amoeba forward. And then on the other side, you're going to have actin shrinking. And what that is going to do is it's going to push the cell forward. So let's watch this video here. So let's watch this video. You can find this on YouTube. So here you can see on one end, you have all this actin growing. So the actin is pushing the cell forward, right? In these false feet called pseudopods. In the meantime, the actin is breaking down over here. So it's depolymerizing over here, but it's polymerizing over here. And that allows this entire amoeba to be pushed forward in one direction. So again, cells move because of actin growing and shrinking. So microtubules are the last category of cytoskeletal protein. And microtubules are the tracts on which cellular materials are transported. Microtubules, just like all the other cytoskeletal proteins, also provide support for the cell. Microtubules, abbreviated MTs, are made of tubulin protein dimers. So tubulin is like an alpha tubulin, beta tubulin. You don't need to know that much detail. But the microtubule itself is made up of a ring, like a hollow cylinder, of these tubulin protofilaments. So it's actually 13 of these tubulin protofilaments in a hollow cylinder make up a microtubule. And these microtubules grow and originate from the microtubule organizing center. And in animal cells, we call that the centrosome. So animal cells have a structure called the centrosome. It's right outside of the nucleus, toward the center of the cell, the centrosome. And the centrosome is what can form microtubules. It organizes microtubules. It contains two centrioles. So there are two centrioles within the centrosome. And the centrioles form the basis of structures called basal bodies that help cells form cilia and flagella, as we'll see. So centrioles are what help form microtubules, cilia, and flagella, right? So centrioles are very important also for cell division. We'll talk about centrioles later because centrioles make microtubules that will help pull apart chromosomes when we talk about mitosis. So centrioles themselves are actually made of microtubules, and they make more microtubules. And again, centrioles are found within the centrosome. There are two separate ends to microtubules. They're unequal, the two ends. Therefore, we say that microtubules are polar. They have polarity. The end that's attached to the centrosome, right? So here's the centrosome containing our centrioles, and here are all the microtubules. The microtubules are attached at their minus end. Again, it's not negatively charged, we just call it the minus end, right? The minus end is attached to the centrosome, and the plus end is toward the outside of the cell membrane. So the plus ends are the free ends. And microtubules can grow and shrink at the plus end. So they're very dynamic. So I just want to show you, this video is not working now, but the microtubules are always growing and shrinking. They're not just staying still. So you can watch this video by using this link up here. The microtubules are growing and shrinking all the time. They're not like train tracks that just stay in one place. So that brings us to the next question. How do materials get transported throughout the cell? So we have two major groups of motor proteins that can actually travel on microtubules. So kinesins are one motor protein that can only move toward the plus end of microtubules. Dyneins are other motor proteins that only know how to move toward the minus end of microtubules. But regardless, let's just think about what these motor proteins do. Here's a microtubule, right, in green. And the minus end we know is closer to the centrosome. The plus end is farther out toward the cell. Suppose I need to get some cargo, some cellular material from one end of the cell to the next. Motor proteins can carry along cargo. So this motor protein, this is a kinesin over here, is carrying along some material. This could be a vesicle containing proteins. This can even be an entire organelle. And these motor proteins travel along, walking along on microtubules, carrying these cellular cargo. Dyneins do the same thing. They just walk in the separate direction, right? Dyneins go toward the minus end. So this is how organelles get transported within the cell. Motor proteins physically attach to organelles and walk along microtubules. Those are the train tracks. Also, neuron signals are sent on kinesins. So kinesins can bring along packages of neurotransmitters to the ends of a neuron. And then those neurotransmitters could be released into the synaptic cleft so they could communicate with the postsynaptic neuron. So I want to take a close look at kinesin. So you can look at this video right now to see up close what's going on. So here's a microtubule, and here is a kinesin motor protein. And it's carrying a huge vesicle of proteins in it. And step by step, this kinesin motor protein is walking on microtubules, bringing along this cargo. And that's drawn to size. This is actually accurate. So this little kinesin, right? These kinesins are walking inside of each of your cells right now, right? So try to visualize that. Each of your cells has microtubules with these kinesin motor proteins traveling along, carrying their substances that need to be delivered. And this happens because of ATP. So it's ATP is required for each movement. So just briefly, I want to show you how the kinesin steps. This is a kinesin over here, and this head has an ATP attached to it. So when ATP is used, then it can take one step over. So it's going to take one step over. Good. And now this one has an ATP bound. So now this ATP will be hydrolyzed. That energy can be used. And then this can take one step over this way. So step by step, this is how kinesin walks on microtubules. Cilia and flagella are also made of microtubules, and those are responsible for cell movement. So cilia and flagella are extensions of the cell membrane that contain microtubules, and motor proteins like dynein help power the motions of cilia and flagella using energy from ATP. So here, like flagella have more of a propeller-like motion. Cilia have a back and forth beating type motion. Cilia are short hair-like projections containing microtubules. They're really good at moving substances along cell surfaces. So cilia, not like in their human respiratory tract, they help sweep any dust particles up and out away from our brain. Like in our nasal tract, we don't want anything going up our nasal mucosa. So our upper respiratory system consists of ciliated epithelial cells that help rush away any dust or pathogens, down into our digestive system. Also, of course, like a single-celled paramecium relies on thousands of cilia that allows it to swim in water. Flagella are long whip-like projections that contain microtubules as well. So these are more like tails. Sperm cells in many species, including humans, have prominent flagella. So here's a quick video. Let's see if this is going to work. I'm just going to open this video in YouTube now. So here we can watch a video really quickly to show flagella and cilia in action. So here is an algae, a Chlamydomonas it's called, And you can see a little faintly here, these are two flagella. I think up here you can see a little better. You can kind of see these two flagella that are moving around and moving to help propel this, this algae toward the light. Here's even better, you can see. So this is helping them find food and get around. And let's look at a paramecium. So here's a paramecium, and we can zoom in and see all of its little cilia beating. So those are the little cilia. Again, they're made of microtubules, and these are allowing the paramecium to swim. So again, microtubules that make up cilia and flagella enable cells to move. So while cells definitely do move, oftentimes within an organism, cells need to stick together. So cells actually need to make junctions with one another to work together. So cells can either form junctions with each other, or they can connect to what's outside of their membrane, called the extracellular matrix, which is usually a lot of proteins and other small molecules outside of the cell. There are three main types of junctions we'll talk about. We'll talk about tight junctions, then anchoring junctions, and finally gap junctions. So animal cells stick together through tight junctions. So a tight junction fuses animal cells together, forming like a watertight barrier, almost like sewing machine stitches. And these are really important for cells that connect to each other that line the digestive tract. Right? We want to make sure that fluid doesn't leak in our digestive tract. We especially don't want like stomach acid to leave our stomach, because that can actually break down and digest some of our own tissues. So tight junctions make sure that cells do not allow any liquids to pass between them, almost like sewing machine stitches. In anchoring junctions, we have animal cells connecting to its neighbors, so it can connect to adjacent cells, or it can connect the cell to the extracellular matrix outside of the cell. So proteins at each anchoring junction span the cell membrane, right? They go through the cell membrane, and they link to each cell's cytoskeleton. Right? So if this is one cell, and here's another cell, they're anchored from bone to bone. This is almost like saying if I wanted to attach two people together by attaching their skeletons, like I would take one bone from another person and attach it to another bone from another person. That's how tight they are together. So it's attaching one cytoskeleton of a cell to another cytoskeleton of a cell. Finally, gap junctions allow cells to communicate very rapidly. So gap junctions are protein channels, almost forming a tunnel, so that materials can go very rapidly between one cell to the next. So this is how ions and small molecules are rapidly exchanged. This is really important in the heart, for example, when all the heart muscle cells have to contract in coordination. So this has a lot to do with ions traveling in the right patterns throughout the right cells, but in synchrony. They all have to be delivered to the right cells at the right time. So these secret tunnels allow those ions to get to the right places at the right time very quickly. Right? Otherwise, they would have to find their way through all of these cells, and that might take too long for the heart to contract. In plant cells, we have what are called plasmodesmata, because they don't have these other kinds of junctions that we've spoken about. So plasmodesmata are channels that connect adjacent plant cells. And again, these are just like gap junctions. They're little tunnels in the cell walls. So these plasmodesmata are little spaces in between that allow materials to be transported, especially important in parts of the plant that conduct water or nutrients. So plasmodesmata are more like gap junctions. And here's a summary, again, of everything we spoke about. And we've spoken about generic animal and plant cells, but each multicellular individual has a variety of cell structures and functions within them. So a neuron, right, has the same basic organelles as a muscle cell, but a neuron is highly specialized for its function. right? So in multicellular organisms, cells divide up the labor when they get together to form tissues. So remember, within a tissue, we have different cells working together. So different cells have to take up different structures so they can work together to perform the same function in a tissue. All right, same idea as in plants. Leaf cells have chloroplasts that can do photosynthesis, but underground plant tissues like roots do not, right? So multicellular organisms divide up the labor and become very highly specialized. So I'd like you to watch this video. It's very important. It's the best video I could find, actually, that summarizes every organelle one by one. So this is a required video to watch. And again, it's a nice animation that goes through everything we spoke about so far. You can also watch the video contained in the ebook on Connect. And this is a closer look about what's really going inside of the cell. I'm not going to go through this right now with you. I'd encourage you to watch it on your own time. These are some beautiful animations of what it's really looking like inside of the cell. So inside of the cell, here's another kinesin molecule moving. Proteins are always moving around, and the cytosol is packed with proteins. So it's not like this fluidy space that's just open, right? Things are always shaking and moving just by random motion. So it's a nice video to watch. So to summarize, we said that cells all have DNA that encode proteins. They all have ribosomes. They all have a cell membrane. They all have cytoplasm. Cells may be prokaryotic, in which they would belong to domain bacteria or Archaea, or they may be eukaryotic. Eukaryotic cells contain organelles that include nucleus, mitochondria, chloroplasts, and they also contain cytoskeleton that consists of actin, intermediate filaments, and microtubules. So that is the end of chapter 3. And next time we will discuss a little bit more about energy and how cells require and use energy.